Why, hello there.
Hello.
Hello there. It's quarantine time, everybody. That's right. We're all going to be kind of hanging out in our houses for a little while for the next few weeks. And you know what we're going to do for the next few weeks? We're going to solve a really big problem, or at least we're going to try. What's the biggest problem right now?
Coronavirus.
And what are we going to do about it?
Quarantine.
Well, what are we going to do after the quarantine? What happens when everybody starts having to go around outside and everything and the virus is still alive? Isn't everyone just going to get sick?
Yeah.
So we have to find a cure. Right. Now, here's the beautiful thing about 21st century genomics and us acting as a global society. Information is abundant. And thank you to the NCBI. I can't even remember it's like the National Biometrics something, whatever. Point is they have the genome already sequenced for many different iterations of the coronavirus. I have written some extractor code, and I'll provide this file to anybody who wants to download it for free because everything's free right now. We got to solve a problem. It will unwind the DNA sequence that comes off their website and put it in an order. And this is a file that I worked on many, many years ago. And then I stopped working on it because it was too hard. I moved on to other things. And now I'm kicking myself because now we've got to whip it out and get this thing solved right now. So what we have here is we have the coronavirus, the SARS-CoV-2 virus, as extracted from a patient in California. So this occurred in the US. There are many different iterations of the virus. It's going to mutate in different ways. And these different base pairs here are going to come out in a different order. So the four base pairs, the nucleotides of DNA, do you know what they are?
No.
They are four different main letters, and RNA actually is what is inside the virus. These are the letter designations. A stands for adenine, cytosine, guanine, and thymine, I believe. Okay? So within the string of RNA, which is the virus, which is about 30,000 of these characters long, what we've done in this pivot table, and what we'll be working on over the next several weeks as I do more videos about this and figure out what this all really means and how we can use it to defeat the virus, is to see the concentration of these pairs, because what they do is the nucleotides end up giving instructions in the cell, and as it hijacks the cell, making more of itself, and also making strings of proteins and different receptors. It turns into shapes and things. And a lot of people have already modeled this, but we need a more just novel way of thinking about this problem right now because we need to solve this fast, and we need to come up with treatment options as fast as we can to save economy and food shortages and a million other different problems that result from all of us being quarantined and not going anywhere. So what we can see in this version from California that's in here is that this is predominantly A's and T's. It looks like there are like the A's to T's look like to be about 2 to 1, that there's more A's and T's, which A bonds to T and C bonds to G in the spiral double helix of DNA. So we're going to start to see amounts of it in here, and then we can also see orders and character sequences. And so one of the things that we worked on previously was different types of chains kind of mean something. Like TAA-CCC can mean something at the beginning of your genome in the human genome. All these things are going to mean different things, and there's infinite number of, yeah, I know I've lost my kid and I've probably lost you too, but this is important for those who want to mess with these files and start to work on how we're going to diagnose how to attack this virus. Here's what I'm trying to say is there are different character sequences, and I've mapped out 3s and 5s and 8s. So every 3-character sequence looks like this when you bring it into the table. If you want to see which one of these 3-character sequences exists the most, you can sort this by the counts that exist. And you see, for example, that the highest frequency in this genome is a TTT. There's 1,000 TTTs, and then there's AAAs also, which is kind of the inverse of each other there. A's bonds to T's. TTA, TTA, and then things really start to drop off. And you don't see things like as many hardly any CCGs or CGGs or GCGs. It's much less. That's going to mean something. It's going to relate into different types of proteins being created. You can also say, well, I only care about maybe the front of the genome or the back of the genome. Well, you can do that by filtering the order. You can do that a couple different ways. Like what these things are slicers, which show the order, the string order of the DNA as it was sequenced. Like character 1 is an A, character 2 is a T, thymine character 3 is a T. All right. And we can open up the slicer and show all our different orders and highlight different areas to see the compositions this way. Like, this is one way to do it. You say, all right, well, what exists in the first 100 base pairs? You can just highlight all this. And you're going to see that in the first 100 base pairs, this is predominantly, it's pretty well distributed, but there's a lot of TCTs and AACs and TGTs. And you can also do this not just for 3-character, but for something like 5-character sequences or 6-character sequences or an 8-character sequences. It's endless. And you can resort this by 5-character sequences. And you see that within the first 100, there's a lot of blank, but there's also this G8 TCT. It's really not a good way to look at this, actually. You should probably look at it in different manners. Point is, there are going to be a lot of videos as I expand upon this and convert this into what types of proteins get created where and comparisons of different mutations or iterations of the virus that already exist in the wild to see, for example, if something like what's going on in Italy or Iran has a different, more aggressive, violent strain that looks a little different when it comes to its genomic order and maybe why that is. There's so much that we can learn right now. And during this period of time where we all have to hang out and really do nothing if we're going to be responsible, we can all spend time focusing on things like this, because once we can come up with some type of treatment option that I believe creating something that will bond to this virus to remove it, to prevent it from bonding to your vulnerable ACE2 protein cells. I think if we can get this to bond to something else through intake of either a pill or a nebulizer or something that gets into the area of the system where the virus is free floating, if we can get something in there to bond to it and pull it out of our system, I think that's going to be the way to go. So we're going to just have to figure out how to do that. Do you have any questions about any of that?
No,
Does that sound perfectly reasonable and not?
I don't know.
No, of course. Nobody understood anything I just said. But some of you virologists and geneticists out there will understand the value of a program like this. So I'm going to be putting it up to download, obviously, for free, and I'll continue to update this day by day because this is a $1 trillion problem right now. If you can come up with a cure for this that can be administered to people after they have contracted it, not a vaccine, but a cure that can help them not have their lungs deteriorate, you've got a $1 trillion idea. And you also move the economy back to a stasis mode that is much more convenient for those of us who would like to live outside of our houses, right? It gets pretty boring inside all the time. I think it does. All right, we're out for now, but we'll be back tomorrow. You have anything to say to anybody, kiddo? Babies, perhaps?
Subscribe.
Yeah, subscribe, sure. Why not? Doesn't matter.